Yes.
Cool. All right. Hi, everyone. My name is Leo Blondel. So I'm a fellow here for those of you who don't know me. So this is just a quick slide to just say that I have many different interests. Some of which are scientific, and this is what I'm going to mostly focus on here. But I've also worked with open source and open science, and I've co-founded Just One Giant Lab for those who might be interested in looking at this as well. And I do a bit of art and art science, and I try to play with data and make data beautiful from time to time. But so today I'm just going to talk more about my current research project here at the CLIF. Which is titled Diving into the Fourth Dimension. But before I do that, I'm gonna give a brief sort of summary of what I've done in research in the past. And so my research studies biological systems most of the time. And biological systems are a complex system, right? If we take an example of a single gene, which will make a protein, that protein will then go on and regulate a lot of other genes in the genome, which will then in turn on other protein, modifying this particular network and creating a complex system. And if we look at other scales and we look at the scale of an organism, we see the same thing with different signaling molecules that will cross-talk between different cells or different organisms, creating a complex network of interactions that will then go up to a particular food chain, for example, where you will then have organisms that will integrate into an ecosystem and have different relationships between each other. And so at all of those scales, right, we see a rise complexity. And this really is the main topic in terms of biological study that I focused on, right? And when I started my research, it was already sort of well into the era of big data, where we've had access to sequencing technologies that allowed us to have multiple thousands of genomes and transcriptomes, but also phylogenetic tools and a much better understanding of the evolutionary relationship between organisms, as well as massive databases online to annotate what we would find in a genome, and protein-protein interaction network and other types of network systems that emerge, which allow us to understand in context the results that we could see with other tools. This also is the era of many types of experiments which allow for extremely rich data, such as microfluidic devices. And in terms of microscopy, we've had many different revolutions that now generate very large, very rich data sets. And so within this, right, I've asked many different types of questions using different types of approaches. The first one, which is the origin and evolution of a particular gene, which is essential for the reproduction of insects, which is called OSCAR. The second one, which is a more network science approach of protein-protein interaction analysis to try to understand the reproductive capacities of Drosophila. And then what I'm going to spend a lot more time on is the analysis of the germ layer specification in a crustacean called Porhalli hawaiiansis, and the use of virtual reality for the analysis of those type of volumetric data set I just talked earlier. So to start with, very quickly, I'm going to go over the origin and evolution of OSCAR. So those are published studies, and I worked with a master student called Savandara Bess, which did some of the work. What we did is we studied this gene OSCAR, which has a very simple structure with two domains, Lotus and OSK, and had very few understanding of its evolution and its origin when I started my PhD. So what we did is we created a massive survey where we collected all of the genome and transcriptomes that were available online. And then we created a database of 369 ortholobes. What we first showed was that OSCAR is a gene that is restricted to insects. And a bit more specifically, it's restricted to this particular branch right before the evolution of Zygentoma, which is the silverfish, which are not fish. But thanks, English. So in that case, that means that Oscar was a new gene. So what we looked at is how did Oscar arise, right? Because new genes, they don't form from nowhere, they need to have a mechanism. And what we found and showed was that Oscar actually originated from a horizontal gene transfer, but more interestingly, it's not just a gene transfer, but a gene transfer that had then a fusion between both domains, which is actually kind of a novel mechanism for gene formation. So we showed a new way for genes to form through fusion of foreign DNA into novel formation of genes. Then we looked at its evolution and how it acquired its function. And so for this, we just focused mostly on its dimerization properties, its properties in terms of binding RNA, and in properties in terms of binding another protein called VASA, which is essential for this reproductive mechanism that it creates, right? So I just went really quickly over this because if you're more interested, we can talk about it later. But I just wanted you to know that this is part of what I've done. And then I'm gonna go from like single gene study to gene network studies, where another study that I did was topology-driven protein-protein interaction network to analyze and detect genetic subnetwork that regulates reproductive capacity. And so reproductive capacity, for those of you who don't know, is the number of offspring a particular species can give, an individual of a particular species can give rise to. So for a fly, it's in the hundreds. If you take a honeybee or an ant queen, it's in the tens of thousands. For human, it's less than 10, right? And so that's basically a very important factor in evolution and ecology to try to understand the capacity of this organism in a particular environment to reproduce. And so to look at this in insects and in other species, right, one of the main factors that regulates this capacity is the ovary, because the ovary is the egg production unit of insects. And so what we did is we made this large screen of all signaling pathway genes to try to understand what were the genetic regulatory mechanisms that would underlie the formation of ovarioles, which is this egg production machinery in insects. And so when we looked at this, we actually found that signaling pathway genes, which were our first target, because that's usually what would drive particular developmental mechanisms in biology, were not a good predictor, because most of them were not enriched in any of the things we found. So instead, we turned to a network approach where we took the protein-protein interaction and we tried to find regulatory modules, submodules, by taking all of the genes that we found that had some effects and trying to find connector genes that would make those into a module, so a connected component, right? And so we took all of the predicted genes that would connect those other genes we already had found, and we measured their effects on the reproductive capacity. And then what we found was that this methodology was actually much better than targeting through signaling pathway genes. And we then went on to do a lot more network analysis of those particular modules, right? All right, so this is what I did in the past in terms of other types of study, and I wanted to just present so that you guys know a bit more where I come from. But then this is more my current focus, which is from like organisms to embryogenesis and the study of embryogenesis of this particular organism, which I've studied a lot, called Porhalli hawaiiansis. And so what I looked at was the germ layer specification in the early embryogenesis. And so Porhalli hawaiiansis, for those, probably all of you who don't know it, is a tiny amphipod. And so amphipod are those organisms you see on the beach when you walk, and they're like called sand fleas often, and they just jump towards the water. And so this particular organism has an embryogenesis that takes about 10 days, and it has a particular property which is essential for trying to understand germ layer specification in the way that I'm going to present, which is an invariant and stereotypical.
Beyond the fact that we know them and they jump in the water, what's the advantage of looking at it as a model?
Yes, so it has, so as a model, it has many advantages. First, it's very easy to maintain in the lab because you need a Tupperware and they'll just survive out of carrots. So it's very practical and efficient. It has a like 3 to 4 weeks generation time, which is pretty short. We can do all genetic modification on them fairly easily. They have transparent embryos.
What do you use as vector for them?
For them, with piggyback transfer, now we can do CRISPR as well. But earlier it was piggyback, which worked really well. And has, so they have really large eggs, so it's really easy to inject them and things like this. But also they have transparent embryos and transparent eggs. And so for all of the development, it's really interesting. And they became the model organism for crustacean, and especially crustacean development, because of those properties. I'm not saying that other crustacean wouldn't have been a good match as well. It just happened that this one was developed through the last 20 years. And so now it's really the de facto sort of crustacean model of development. And why crustacean, why it's interesting is that crustaceans are the sister group of insects. So whenever we want to try and understand the evolution of development or the evolution of embryogenesis in insects, they give us this really interesting sister branch to study in terms of evo-devo, what happened in insects. And it has one more property, which is this invariant and stereotypical development, which is really, really interesting in terms of how you can understand development in an organism, because by the eight cell stage, right, you already know from each of those blastomere the descendants in terms of tissue type and germ layer that it's going to give rise to, right? So for any analysis, you can inject, right, any of those blastomere, for example, and trace with like fluorescent tracers. That's how we actually got to those state maps. But what no one has looked at, sort of until like I started, was what exactly happens between the stages, right? It was like still shots of the fate map, but not the dynamic processes. So what we know is that overall, those blastomere will contribute to big part of tissues, but we don't know exactly what each of the cells descending from this will do and how they'll organize and what will actually happen to form this. It has many other properties, which I'm not gonna get into, but it has very interesting processes for segmentations that have not been seen in any other organisms as well. So the segmentation information is really interesting in this organism in terms of how it generates each segment and what is the regulation of this, which is not completely well understood yet. Okay, so to try to study this processes of this development of this organism, what I did was I took one cell stage embryo and I injected them with tracer. And so those were mRNA tracer where they would express a nuclear and a membrane fluorescently tagged proteins. We would then take those embryos and place them in a LightSheet microscope. So a LightSheet microscope is a particular type of microscope which has the advantage of generating multi-angle Z-stack data sets. So what that means is we can go through the embryo and have those particular three-dimensional but not high resolution like in Z three-dimensional stack, but you can then rotate the embryo and take another movie from a different angle, which gives you all of those different stacks, which you can then virtually register to each other to then go and fuse all of this into an actually isomorphic 3D volume. What this gives you is a high resolution with about like 0.4 micron resolution per voxel data sets. And those very nice 3D images, then the question is what kind of new analysis this data can bring, right, to try to understand development. And so the first thing that most people think about is lineage tracing, right? So lineage tracing is this method where we take the cells from their inception and we follow them through time into the development of the embryo. When we do this, what we have is the identity, position, velocities of all of those cells. And so it's their history. And in development, right, this history is extremely important because we know that gene network will go through different cascades to give an identity, a unique identity to each of those cells, right? So trying to have that type of data set is very important to understand development. But the problem with generating that type of data set is that it's extremely time consuming. To generate this particular one, it took me about 6 months of tracking all of the cells by hand, right? And so this becomes a very big problem when you start to want to actually have statistics. about what is actually happening when you want to compare things, right? But with that type of data, we can still, get into, like the dynamics of cell division, like what's the rate of cell division. You can also get at the velocity of each of those nuclei, right? And you can do this sort of like broadly at the embryo level, which is sort of interesting, but not that interesting when you start to actually map what's happening with the lineages and the different tissue types, right? Then you can start to separate subgroups and look at each of those subgroups and if the velocity or division rate or their position, their neighbors, what exactly does that change? And this allows you to much better understand the development of this organism, right? And this is sort of what prompted me to go towards virtual reality, because those data sets were natively three-dimensional and they required a large amount of manual annotation, right? And this manual annotation on a 3D object on a 2D screen is extremely tedious and extremely hard to actually perform and do well. But before I get into this, I'm just going to give you a quick history of virtual reality. So VR as a medium, we can say that it started with stereoscopy in the 1900s, but it was still images. So the first moving virtual reality images really were the Sensorama, which was created by Morton Heilig in 1956, which was this sort of personal movie theater where you would put your head, and it was IMAX 5D at the time because you had like wind coming and like the thing would shake, right? So it was like really the invention of like today's modern cinema already in a box. And then he went on to propose this design of the first sort of head-mounted device or HMD of sort for VR, which he never built because his design was completely impossible to make, but was then sort of taken by Ivan Sutherland with the Ultimate Display in 1965, which was the first time that someone actually plugged a computer with some sort of rendering engine to see in 3D and with like PTO, like this capacity to move around something was like, really early days devices which would fit on your head in a weird manner. But then, so like, after this, you have a lot of development and a lot of interest, mostly in the army, for simulating, simulation of planes. And the first sort of like democratization of this system is by John C. Hart and its team in 1992 at the ACM conference, where they presented the cave system, right? And so the cave system is this idea where instead of having the mounted device on your head, you will project through all the screens around you. You'll create a box, video projecting box. And so through that box, you will then wear glasses that will, like do right, left eye with like either active or with polarization of light. And they'll just recreate a three-dimensional images, but in a particular environment. So you're moving, you're not reprojected in an immersive environment. The immersive environment is around you, right? And so those cave system is really what a lot of research from the 90s to today have focused on in terms of virtual reality, right? And after this, you have the invention of, consumer VR HMD. And this is much more recent, right? It's like 2017 is really the first time where we see this enter the market with, the Oculus Rift and the Vive and the Valve Index. And then like today you have these like 8K screens and like it's really exploded in terms of adoption and in terms of technology and affordability, right? Because the cave system is very, very expensive versus a headset is just in a couple $100 today, right? And so with VR, what sort of has been the main three topics of research so far, beyond the army and military stuff, is neuroscience and
95% of the market, which is 95% of it.
Yes, which is the vast majority, but we don't know about it. It's mostly non-published. So, so it's neuroscience and behavioral science, and this makes sense, right? Because suddenly you can trick the mind. With virtual reality, there's this effect where you will become embedded into the system, right? Like you will reproject yourself in a particular environment and your brain will believe it, right? I mean, I'm sure you all seen the videos of people that you push when they're standing in VR next to I don't know, like a massive drug, and they're just going to collapse on the ground and just like be extremely afraid that they actually might have fallen, right? This effect, right, of immersion is what allows neuroscience and behavioral science to use VR for many different things. When you take a mouse, you can put it in a maze. When you take a human, it's much harder to put them in a maze. But in VR, you can actually do those sort of maze-type experiments. But in psychology, it's the same. Like, this is one of the main use of VR is for like treatment of PTSD or phobia, where it works way better than anything else, right? Because you can then subject the user to fear in a safe environment. They can always remove the headset, right? You can stop the simulation, you can stop it. And so through small induction of the trauma, you can really have very curative effect in psychology. The next sort of like main field is education for in VR, and how does having a reprojection help with education, or at least changes education, right? With questions such as, is it better to have the teacher next to you, in front of you, which reprojects a whiteboard with a mirror in front of you? Or do you have like a better teaching experience when you're face to face and talking, or with many other hundreds of people? Those sort of question in education are very interesting. And the main field for this is medical sciences, where you also have training for surgeries and things like this that can happen in VR, where you won't kill the patient because the patient's not real. And then the last one is data visualization, because this medium allows you to reproject into a 3D environment. And so you have this 3D perception and completely new way to interact with data, right? And this is really where sort of like my research stands in, mostly, was maybe a little bit towards education, a little bit toward, neuroscience as well, but like mostly into like, how do we see those embryos and those tracks and all of that type of data, which is three-dimensional in time by nature, in a better manner. And so in 2017, I sort of created a prototype VR application, which was a VR viewer where you could take those microscopy data sets, you could open them, you could just manipulate them in terms of like intensity, and you can play those movies and switch through the different channels, right? And so like sort of this is where, like I stayed for a long time until today, where I entered a decree to continue on those research and go more towards, research questions of what does seeing in VR, those types of data sets actually use. So the questions I'm interested today are, what is the effect of virtual reality, right, on intuition, insight, and the analysis capacity of the researcher? Because this is sort of a new field, and it's not completely clear that putting those data sets, those microscopy data sets in a virtual environment would actually increase or decrease the intuition and insights that you gain from the data. And this is extremely hard of a question in terms of actually measuring intuition and insight in a researcher, because you don't have a really good control, right? Like it's not like I can say, oh, like I'm going to show you this data set and then I'm going to put you in VR. Like, do you have more insight now that you've seen it in 3D? But what's a bit more easy to at least measure is the analytic capacity of, the sort of natural manipulation of labeling and interaction with data sets in 3D in VR, because your hands are there, and when we exist in the 3D space, and when you can touch in VR, then, can we actually show that this increases speed, ease of use, and those are questionnaires that we actually can design much more easily. And the second part of the research that I'm interested in is to look at artificial intelligence. And what happens when you actually have this visualization capacity in VR and you start to plug AIs into the mix, right? What happens when you dialogue with a machine learning model, an artificial intelligence agent that will make prediction and show you those predictions live that you can then give feedbacks on through data labeling or other corrections, and that will just create this loop of model selections and intermediate results, right? And this is sort of dear to me because from a computational biologist perspective, I've worked with many biologists who do not understand black box AI and machine learning agents, and creating those systems where you are seeing what the machine learning model thinks can bring an increase in confidence, right? And this increase in confidence is very important because most researchers need to understand what the data is, right? Like what are actually these models? What is happening when you're using this particular artificial intelligence? Because if you don't understand it as a biologist, there's no way for you to be confident that this actually did something right, right? What if something wrong happened? What if the prediction is wrong, right? Like if you don't understand what's happening in the model, you can't really use this for your research. And so doing those sort of like AI in the loop model is something that I think would be very valuable and interesting to researchers, especially in biology, which lack sort of the knowledge to understand AI. And so how do we do this? So how do we get there? And how do we sort of like ask and like start to answer those questions? So the first thing to do is to design an extensible sort of VR microscopy platform, right? And this is what I've been doing for the past 2 months, trying to work on creating this tool set that would allow for all of those sort of studies to be run and for biologists to be able to start to interact with big data into it, right? And so for this, I have a developmental plan, which is made of basically 3 phases and different items. And so the first phase, which is the one I'm focusing mostly on for now, is to create a platform using open source, mostly like tools, and releasing it as open source, which would allow for multi-user interaction to study 3 types of datasets. And the reason those 3 types of datasets were chosen is that I'm focusing on LightSheet microscopy in the context of embryology and morphogenesis. And so those are 3D plus time images, the sort of volumes that I've talked about, but object tracks, which are those cell tracking, and image masks. And so image masks are, I haven't talked about them yet, but they're this basically selection, pixel selection, where you would select where in a picture you have a cell. Those are essential because when we start to want to use AI, those are the sort of tools that we need to do and to create to interact with artificial agents such as segmentation, which will find those cells in your data sets, and tracking, which would then track them. And so for this, we need to create the tools to be able to observe, track, and paint those masks, right? And so phase 2, which will probably be after this fellowship, would basically be to integrate artificial agents with that mix, right? And so for this, there's tracking and tracking AI and segmentation AI that sort of already exist. And then finally, in phase 3 is like open this more with an extendable code base to add for plugins for the community to be able to generate more tools, more analysis capacity into this data set, right? And so I'm not sort of starting from nowhere. There's a lot of things that were done already. So for everything that's multi-user and everything that's VR development, Unity is a system that already has a lot of those tools created. For tracking and segmentation, there's 2 pretty good algorithms that would be extensible and could work well in this system, which is the Baxter algorithm and Stardust. And for 3D image and object tracks, there's the entire Fiji ecosystem, which I can take the file formats and I can take the methods to be compatible and backward compatible with the entire sort of analysis pipeline that is already used by people. And so the next steps is going to be finishing this this platform, but then to conduct some other user studies in terms of measurements of the classic sort of HCI parameters, such as the time of completion, the ease of use, the understanding of the interface, right? And this I hope to do towards like May here. But then also the value in teaching, which is something that I would like to do because I want to try to get at this intuition and insight question. And to get at this, where I sort of devise an experiment which could allow a bit more controlled environment, is if you actually try to teach a complex 3-dimensional process, such as gastrulation, which in the embryo is the formation of folds at the beginning of embryology. If you show 2D movies of these processes to students, but then you put them in a VR environment and you show them the same, right? and then you ask questions at each step, and then you reverse the process, the order at which you do it, and you try to measure their understanding, their actual complex understanding of that process. Does VR actually bring something to the table, right? Like, does VR help them understand better a complex three-dimensional phenomenon? And so for this, I'm working with the SCORE students on sort of like questions towards this. So we'll see how far the group gets, but I think it will be quite interesting. And then finally, and this is sort of more of an open question, which I'd love to discuss more after this, if you have ideas on how to get at this, is really how do we measure intuition and insights in researchers, right? How do we see whether or not VR increases the intuition and insight capacity of researchers using such tool? And so, at least for me, what I want to get at with that sort of tool is to get at large-scale analysis of those types of data, right? I'm hoping that this will reduce from 6 months to a few weeks maximum the analysis time and the cell tracking time of a particular data set, so that we can start to do larger scale analysis, such as comparing 6 wild-type embryos to 6 ablated or like conditioned embryo, which data I've generated, but I can't spend, you know, like 3-6 years just tracking them by hand, right? So to get at this capacity, we need tools like this, which currently don't exist. But it goes beyond Porhalli hawaiians because it also gets into morphogenesis and the study of morphogenesis in general. But not just that, like if we take collective behaviors such as bird flocks or fish flock, those are also three-dimensional and time processes, right? So it's not volumetric data per se, but there are three-dimensional, like 3D cameras that can, you know, record those bird flocks, and people have done it. But it's the same problem. You need to track by hand, and you need to try to understand it. But if we start to look at this in VR, you know, maybe a researcher would be able to understand or develop through plugins like more interesting analysis capacities with those systems. And that's for now what I have done in this VR field. I just gave you one more question. I made like a half an hour talk.
Let's start from questions from home, from people who are not here. Bring people, any questions?
I have a short question regarding the study about reproductive capacity. Didn't notice, was it published?
Yeah, it was published, yeah.
Okay,
It's
so
in eLife.
I'll ask you for the reference later. Looks really cool and great talk altogether.
Yeah, Roberto.
You said Roberta?
You.
More of a question. At the beginning of your presentation, you proposed a view, you know, starting from the genes to the proteins to the networks to the organism, etc. That's very, very gene-centric. I think it would be interesting probably to consider some less gene-centric approaches to embryogenesis and morphogenesis, and then, you know, you're very welcome to come for a coffee, or I can just one over there, because I would love to pick your brain on that. And I think in particular that the type of view that you are proposing, it's targeting a level which has like an independent sort of causality that's not the genes. So, for example, I don't know, when if you think the Chaldy plates, you know, when you have dust in a plate and then you push a violin or a bow and then you make a shape, the shape is not in the bow, right? So to understand the origin of the shape in that very simple case, what you need to understand is the system. It will not be the genes which are probably triggering, you know, providing the trigger for the shapes that appear. And what's quite unique to the type of tool that you are proposing here is that it lets you understand better this other potential source of causality. And there's a series of tools that you can try to, would be super interesting to try to add to understand, for example, the mechanics of the complex system that it's being implemented there, no, like how the shape of the organism, and I'm thinking on Growth and Form, Darcy Thompson's book,
Of course.
like how that type of phenomenon could be understood through tools that allow you an intuitive perception of these patterns that emerge kind of autonomously because of the complexity of the system.
No, and I didn't, I mean, I made this sort of red line like this, like from genes to organisms, right? Like, because it's, I think it's nice and it's still nice to think about it, but I don't want to imply that we need to have a gene-centric vision of biology always, right? And what I started to like, the very beginning of the talk was really like, there is complexity and complex system at each level, right? So you need to study those complex system at each level. At the end of the day, dude, like we're still regulated by genes and genomes, right? So like you can sort of go back to the gene sort of always in biology, which is like, you know, for the good or the bad, right? Like it doesn't mean that we don't understand, we can't study or understand causal system at the cell level and look at how cells interact as they're individual, right? And it's super interesting and fascinating to me to like look at this that way as well. And I'm more interested in like the complex system and what emerges, like what's the emergence of those properties from the system as like small bits and parts, more than trying to get at what is the gene that's causal for this, right? Yeah, and I mean, that's the whole like Oscar story, for example, is really more like, let's take this gene, which is the single gene, but actually let's look more at its evolution and let's look at how it sort of changed in time more than how it functions in its nitty-gritty detail in a cell to do something, right? Yeah. Yeah, let's grab a coffee, Alberto, in any case.
I'll go for you. So no, I was about how to measure the intuition and insights people have. You might have seen the hidden gorilla paper, where they like gave students a data exploration task. And like some had an hypothesis before and some didn't, the ones that didn't felt that there was like, if you plotted the data just as a scatter plot, it showed a gorilla. So maybe there's like a similar approach for you to like, there's something in the data and you see how many people find it using traditional methods versus VR to say, these are common artifacts or something that people might miss and do they do better in VR compared to, which is not traditional usability and it's maybe also not really intuition, but it's insights people might have missed.
Yes. Yeah. No, and that's interesting because like when, like personally and other people who are working in also like, you know, the field of VR and microscopy, like it's something that every time we talk, we realize like it's the same thing. Whenever I took someone in VR and the VR tool that I made back then, and I just took a lot of people's microscopy data at Harvard and I would just like show them their microscopy data in it. Almost universally, the effect was, wow, I never saw my data that way. Like, it's super interesting. I don't think I understood the processes in this particular manner, right? Because suddenly it's actually three-dimensional. To get at this, like, it's you know, like, how do we measure this, right? And so, like, yeah, sure, like, we can make artifacts, like, you know, like, that's like an artificial way to sort of get at it. But, like, this unlocking mechanism is really like hard, I think, to sort of get at.
Actually, I wanted to say something along the line of question. So I think that I think there are different elements in what you're proposing. Okay? The hypothesis that you make, right, is that embedding people within this kind of virtual reality will help them get better understanding of the system. Mm-hmm. I think that, and that's one. The second hypothesis is that if you do this, you pass a barrier not only in terms of understanding, but actually who can understand. Right? Because we are used that only people that have been looking at these images for 10 years and are experts and have a PhD or an engineer, whatever, have been doing it. But actually, what you're suggesting is that it's a matter of intuition, right? And so it's not so much based on prior knowledge as much as our intuition in navigating in space, at least to some
extent. Yeah.
At least to some extent. Mm-hmm. And the third element is that people have a motivation, right? The third hypothesis that you made, right, is that people actually have a motivation to go into this space and look for something. So yes, when you're a researcher and you know what you're looking for, then you're probably motivated if this helps you. So I wonder if now we're trying to democratize this. Okay? So when people were looking at, I don't know, galaxies, is a good example for this, right? In which people do a very, very boring task, right? But because of this romantic idea of I'm going to discover a new star, right? They're going to do this. Okay? So their mission is very clear, right? They are trained on a set of knowns in order to find something that doesn't fit this pattern.
Yeah.
And so the task is very well defined.
Yeah.
And so I wonder how would this translate in your system? Okay, so what kind of, because when you're a researcher, you know your question, right? It's an individual question, right? And that's sort of what makes you an expert because you know to ask the question. Right? Can you translate, so in the databases that you have, right, can you list sort of the list of, let's say, 20 things that are not known?
Yeah.
Right? But that working within the VR or working with VR in an interface with an AI right, should be solved.
Yeah.
Right? So I think that if you make this as sort of, the idea is not necessarily to gamify it, but at least to make specific tasks, not just, okay, let's see what people come up, and this is a part of it is doing it artificially by putting an artifact there to see if they find it, and this can be part of the training, right, that you do. Right, and then the idea is, can you do sort of a training session with some ideas like Bastian was doing it, but then, let people, tackle real research
data
questions, right, and come with insights. And I think that's basically what you can do in this is that you can measure on the one hand, like, are they able to find the artifact better in this system rather than the other? And the other is, can they solve or can they reach scientific knowledge, right? Better, faster, etc. As compared to people that were either not trained in not the VR settings or in your VR settings.
Yeah.
Which means that the questions that you ask have to be well defined, but also questions that people feel as motivated with as finding a new star. Yeah. Right? If they understand that if you solve this, right, some neurodevelopmental problem that exists in humans, right, you are one step better in order to solve this, right, or whatever, right. But any kind of infliction of the results to their own life values, etc., would make this thing. And so these are the things that I'm sort of still missing in the kind of how to make this more than a Leo project or a Leo class, I don't know, where Nadine Pereiras and other people that do these kind of things around us to a massive scope.
Yeah, no, that's very true. But like the problem with a lot of those data sets too is that, sure, like I can think of questions such as like cell motion and cell motion dynamics, which are sort of general enough, and you can make particular hypotheses that you have in the data that people can look at. But then at the end of the day, like each organism is so different, right? And so the development happens through those morphogenetic processes where you have cells that move in particular directions. And it's like understanding this does sort of take this expertise, right? Like to sort of get at to see something in that sort of data set, you need to have the knowledge that there exist those systems of genes that will push limbs growth because like genes will activate and cell. At least I think so. Maybe I'm wrong, right? But like Galaxy Zoo is like sort of easy because it's image recognition pattern, right?
If you show the end of development and where you are now, and you say we have no clue how cells got to this region, and if we understand this, we understand formation or malformation of legs or whatever.
Yeah.
And then people will really focus
yes
on back and forth of seeing where, you know, where this escape from, right, et cetera, et cetera. So make very, very concrete questions that people can, so because when you segment, you know, okay, I'm going to segment this lineage now, right? Because you know why. You want to motivate someone else to do this.
Yeah.
You have to.
Yeah, I mean, so like that's sort of almost the other approach.
Or the system that is really worth doing it. But I think Roberto had another point.
Roberto?
Yes. So in brain MRI, people have been building these beautiful 3D models of brains for like really a lot since the 90s. And you know, they are very, very nice, much nicer than looking at slices, which is what people still do today in the hospital. And it was very, very frustrating for all the people working on that when they would bring their beautiful tools to the radiologists, they would look them, they would say, yeah, it looks pretty cool, but then when they try to actually get the insight on what a patient brain has, they still rely on the slices, you know, because they know, or looking at this disorder, I have to look at the coronal slice that cuts this and that. So the framework is all 2D. So I think that if you try to measure insight or try to see if people are able to get new information, you will also probably need to get some sort of naive people, because if you will ask a biologist that's used already to working or looking to specific views in specific to these slices, they will be completely, you know, they would have to unlearn what they know how to do in 2D to be able to embrace the 3D. Like the case is that even today, like 2020, in the hospital, radiologists still print their brain images and they look at them in 2D.
Yeah.
So it will be important to not only pick the right questions, but pick the right people, like naive people, students that haven't been exposed to the 2D world. And that you know, I pasted this link to, you may know the work of Doug Engelbart in the 60s, you know, when they were defining the new graphic interfaces for the computer. And their project was about augmenting human intelligence by testing it on themselves. So probably by using yourself these tools, it will be you who will probably get the first insights, and then you can try to see if people are able to
reproduce that. Yeah.
It will not be looking what people already see in 2D, can they see it faster in VR, but it's, you know, first using it in a test group where this insight will appear, and then from there create the new questions that you will probably try to test in an evaluation phase.
Yeah, and it sort of gets at what I was just saying, which is like try to define what are those insights like accurately to try to like measure them, and like getting them from myself might be the best sort of way to just at least have a list of them. Yeah, that's true. That's fair. Yeah, no, like I mean I think for the naive part, like, I mean, it gets at what we're doing with the SCORE students in the project that I've tried to devise with them, which is to create synthetic data sets and try to look at synthetic data sets, because then, you know, it's easier because you control everything and you can get at asking questions such as 3D shape recognition and memory, and that's much easier when you actually don't look at real microscopy data. But it also sort of lacks this, it lacks a bit of use case for the researcher, but maybe it will build at least like the ground, for getting in this. So, I mean, like that's sort of one of the avenues that like I'm definitely exploring and want to keep exploring. And there's some interesting papers on this. There was this paper which looked at Jenga style system. So if you take like wood blocks and you just arrange them in a random Jenga position. And then you ask someone in a room to just look at this, and you can just look at it, not touch it, but just look at it from all angles, whatever you want. And then from memory to rebuild the exact same Jenga tower. Then you do this on a 2D screen, and then you do this on a 3D screen, and then you do this in VR, and they did it in a cave environment as well. So with a headset or a cave. And so what they showed was basically like the moment where the person can start to do as well as real life observing the 3D system is when they started able to walk around. So as soon as they had like spatial memory of where their body was when they looked at the 3D object, then their recall increased drastically, right? And so those you can only do with sort of synthetic data sets. And I guess there's like people working on this already, but not much with microscopy. It's more like general sort of like neuroscience of like, brain understanding. Yeah.
So I'm going to jump in.
Yeah, you go
for
first.
my question, and then just because it's related to this, I was also wondering that this jump between a 2D movie and a 3D environment on the screen that you can walk around in versus a full 3D with goggles and everything. It seems, I can imagine, if I can look like this, it feels more natural and you're exploring, but I would imagine that step is a lot smaller than the step of going from 2D to any kind of 3D.
Like spatial 3D versus 3D.
Like what you're doing versus having a thing on a computer screen in which I can walk around like a video game or something, where I can walk through the brain, move the cursor, move the camera that's looking up and down. Which do you have a sense of the difference between that and your VR?
Personally or like scientifically? Because scientifically, like I don't actually have an answer to that.
Okay.
In terms of what exactly is the difference, except the sort of studies of like looking at a 3D object in 2D on the screen doesn't actually improve your memory recall of it from just the flat 2D images.
But I'm talking about the 3D representation on a flat screen.
Exactly, right? Like that doesn't help you much, right? No.
Okay.
Compared to, so like it's really, at least in that study, it's really like the fact of being able to walk around, right? Like physically, like your body, like your physical memory of your body in motion, which triggers that understanding, right? Not the virtual like motion. Yeah. That's just one study though, right? Like in terms of, but like there's something when you look at a lot of, like personally, I've looked at a lot of 3D images, right, on a 2D screen. When you look at the 3D images on a 2D screen, there is this projection, like always, you're always looking at a flat thing. So you always need to do artificial 3D with shading, where you basically put a light, right, in your scene, like an artificial light. And so those artificial shading, they induce artifacts in your own perception of it, right? Because those shadows, they're not real, and so like it generates a perception of the object, which in itself is not completely real. And it's, I want to say like clumsy, because it's not completely clumsy, but you still need to rotate, and so you need to remake in your head a mental projection of this 3D object. And so that actually takes a lot of time and a lot of processing, brain processing, to turn, like rotates, like go around it, or like, either rotate on the screen or like walk around it. And your brain does it. It does it really well. Like it will regenerate a 3D projection, but it takes a lot of effort. Whereas when you actually are in VR, like this effort is just completely gone. Like it is in 3D. So like your brain focuses on something else. It's almost like, I mean, I don't have anything to say this. Like I sort of want to make, I think that's one of the case that could be made, which is that from just a neuroscience perspective, you have a lot less processing to do. And so you have a lot more sort of potential for other types of processing when you're looking at it in 3D naturally, right? But it could be to just put 3D goggles on a 2D screen which projects in 3D, right? Like that could be the case. And I don't know of studies that have done that in terms of looking at this.
So I think I wanted to like go back to like the crowdsourced aspect of it, and I think there's one reason why in VR this is much harder to do, which is first that like there's very few people in comparison to Zooniverse Galaxy, so you can play, everyone can play it from their phone even, which goes to the other thing is like it's like an effort. You need to like want to go to VR. If you have like 2 minutes between meetings, you might go to the Zooniverse website and click 3 galaxies and be done. Very few people will put on the goggles and say, I will quickly.
If you listen to metaverse versus, right, there'll be more and more people that will live in this kind of environment.
Yeah, but it's still like that's like the but the next
that's exactly what I was asking. Can you do the 3D on the screen? Because it's a lot easier, cheaper, more accessible.
Yeah. No, and it's.
But I think that even if you don't have data, just like anecdotally, I fully agree on like the impact of being able to walk around. It would be interesting to compare you being in VR, but you cannot move and you still need to like turn around things, but it's in 3D and it feels 3D compared to you walk around. Because I think like there's like a really big difference to experiencing VR while you sit around like the world passes by you, compared to actually physically moving in a space. And I think there's the biggest.
No,
There's one thing about intuition that I'm trying to sort of figure out, and this is sort of, looking at 3D proteins many years ago, for a long, long time. The anchors you have in your memory are very, very few. Okay, so if you look at the protein, right? You have carbon, nitrogens, and hydrogens, oxygen. You have four different things, right? It doesn't matter where you are in the protein, right? This is what you see. But so one of the interesting things I think is how to render different patterns and how to give, how to encode in VR more than information. Okay, so if I'm looking at the movies of collective movement and non-collective movement, and trying to think about correlation of movement in what you say, right?
Yeah.
If all I have around me is sine balls, right? It's going to be very hard for me to understand, is this the same sine ball that I saw 1 second ago? Or am I following the same one, right? Because I could imagine that in VR, I sort of take one of the cells in my hand, right? And I walk with this cell until it reaches the end of the movie, right? Which could be really great. And it's one way of doing doing the lineage, right? And if the cell divides, then I take it in 2 hands, and I can decide that I can take. But if there is a way that I can see the cells around me with more information, like when I walk in the street, yes, there are only windows, right? But each window has a bit of a different pattern with different threads, then the environment in which I am embedded in becomes much richer, and the chance of getting more information out of it to see, yes, I've seen this guy 2 minutes ago, right? He must be following me, right? Whereas if this guy is just another red ball, I will not be able to see this.
If it's just a sine ball that's among 100 other sine ball, I'm not gonna see it. So like basically what you're saying is like, how do we make alpha helices and beta sheets representation? Because when you look at a protein in 3D, right? Like if you look at the carbons and the nitrogen, you don't see anything. It's a ball, right? And it's really hard to read, but suddenly you schematize it in 3D still. And then you understand it. Like you see the protein structure way better. Like,
Or you say, okay, I have one, I give a label to the 20 cells that I can see around me, right? And this label stays until the end, right? So I can see how far I am from them. That gives you a notion of this. Anyway, there are these kind of things that are sort of artificial, and AI can help suggesting what would be the most recommended things that you should label identifiers for.
No, that's a really good point.
I totally agree, and I was trying to think of something similar. On sort of a higher level, there is data that becomes so complex that by looking at it, you're not going to see anything. Like this is why we do summary statistics and measurements, because just looking at things, we are not as good as extracting patterns when things get really busy. So I'm trying to see where is this sort of the boundary between I need to look at it to get the intuition, or I need to label it really well, or cluster it, or do something to get the intuition, versus maybe my brain is just not good enough to find this pattern. I need to find the right thing to measure, whether it's the speed or something. I don't know, if you look at the genetic code, you can stare at the ACTGs for a long time and not see anything.
No, but like, so for example, one thing that's interesting, like if you look in those movies, like you might or might not see, but there's those like rotation, for example, that happens. You have like two spiral rotation, right? Like the way, like, like I personally like saw those and like started to understand them, and it's actually like cellular rearrangement that is generating like different particular parts of the embryo at that stage. It's like when I was in VR, this is when I saw those, right? Like I didn't necessarily like see them prior to actually looking at them in 3D. Then suddenly in 3D, like you really see those like vortex like processes, and then like you can then go back to 2D, right? Like you can then go back to 2D and stuff like this to like look at what's happening in more detail. But like like even in complexity, you do gain sort of insight sometimes if you're not trying to track a particular cell, but you're like trying to actually get the general purpose and the general process, right? Like this is, I think, one of where this shines a bit more. I think is more in this, is trying to understand general process or like see pattern in this complexity that you might have missed, right? And then it goes back to making artifacts, and it goes back to trying to find like particular insights into that like you would not have. Or less likely.
I mean, also, I would say, I mean, the goal is not to like make this less complex. Like, yeah, like you wouldn't have any cyan balls. As like the whole task for you in VR is to find out which have the same color, like which is the same identical set over time. So I think it's not that you try to like make it less complex, but by moving into VR where you move around the thing, you can your brain can handle more complexity than it could in a 2D movie or 3D object on a 2D screen. It like allows you to handle more complexity than you could handle before. That's in a sense the claim behind this, I would say.
I guess I'm sort of wondering where is then the upper limit of how much complexity you can handle even in 3D?
So, I mean, that's totally my side. One of the systems, the last system that you mentioned is bird flocking, right? I would have loved to learn, right, just by embedding myself into a flock, right? What signals do I need to get in the surroundings in order for not being not touching any bird and being part of this collective, just but embedding myself in this and understanding if this bird is here, right? So many models suggest, right, what are the 2-3 parameters that birds measure in order to
flow and stuff like this.
connected. But if I can understand this intuitively on my body, right, I mean, this would be an amazing experience. If this would be a blockbuster game.
I've got a lot of artists that actually tell me this, that I need to make a version that's you become a cell. And you're the cell, and you're moving through the embryo as the cell.
I thought, you are catching a cell and then following the cell, right? But
You are
the bird,
right?
And I have to make sure that I don't clash.
Yeah. It's like, you need to fly through the flock, and
That's a big part, you want to be part of the flock, right? And when you're really good at it, you understand how your small changes might change the behavior of the flock, right?
Yeah,
Whether it's
simulated.
Somehow the collective decides to turn right and left, etc., because it's some sort of the collective, right? And so, when you master the game, you can decide. You put yourself in the right position where collectively you move people around.
Even the same with cells, to be honest, right? Like if you're in the epithelium in a cell, right? Like you have cell membranes, you have cell pressure around you, right? You can't move anywhere. You need to move with the flow of the cells as well, right? Like it's similar, like, like cells don't move randomly. They still follow signals, even if they can detach, go mesenchymal and like go places, but that's still like, it's.
So basically the question is whether between sort of observing and being part of the dynamics. And I think that maybe you can, so observing, I can understand this is something that you can compare a 3D screen to VR, but being part of the dynamic, right, is something you cannot do in 3D.
No,
right? But maybe that's where the VR has a real advantage. And you really understand different things.
Yeah, that's interesting.
Hey, it would be interesting to give a thought to the very idea of representation. No? I have the impression that when we do VR and when most people do VR, what we try to do is to bring some sort of the space for, I don't know, like you try to be at the Louvre, you try to make everything to look as if you were in the Louvre. So it's some sort of representation that's a very, very like bijective map between Louvre and here, or the embryo as it is organized in 3D, and that may be underusing the possibilities of representation. No, so as Ndule was saying, what are the limits of what we can grasp through vision? You know, our visual system is very good at grabbing some specific statistical patterns in data. So actually what we get in our retinas is just 2D images in each of our eyes, and then the whole 3D world with the distance and depth and even the perception of the properties of the different materials are just made in our brains from these 2D images in our retinas. So probably if it would be like representing the data in a more free-form way so that it will be the best adapted at our abilities of grabbing regularities in stimuli could be interesting. For example, not have time represented through time, but you could represent very well time as another spatial dimension. For example, that's something that people do when you put a 2D image and you make that into a volume. The same way that you represent a 3D volume in a flat screen, you could represent a 4D volume in a 3D space, for example, by projecting 4D into 3D, and then eventually at time. And then, of course, we have our eyes, but we have also our ears, which are very good at capturing some other different type of statistical regularities, and probably thinking more globally at the way in which we can transform the data so that all the maximum complexity can map into our ability to perceive patterns. Could be a very cool approach, to like go beyond data visualization to just data representation very broadly.
Yeah, embedding the data. Yeah, like what do you think would be like more intuitive for brain, right? Like than like general like.
Well, the Gestalt psychology gives a bunch of ideas of ways in which a bunch of independent stimuli can turn into an object. Like for example, when you can have random points moving around, but if they move with a certain coherence, then that brings you a person dancing the tango, whatever. There's an old demo like that, with just 11 points. If they are spinning the right way, you see the person, you see the volume of the person. So there's all these Gestalt principles that would be interesting to explore. What we use a lot in brain anatomy is flattening things in a way that we try to recover different aspects of the data by changing the space of the embedding. But for example, I imagine that some sort of transformation of the 3D into like time data into space would be cool, or if you have the lines of the tracking for each cell, for example, that will make an object that you could explore the time component of all your cells as if it were a sculpture. So have a God's view on the development of your little embryo.
No, it's actually interesting because like this gets at the question of how do you represent the tree, for example, right? Like the lineage tree. How do you represent this in 3D, right? How do you make a useful representation of this lineage tree, right? And it's true, like, I mean, you could reproject that 3D object, like, because they're spheres, so it's easy. But you can make like a, Mercator style or better style prediction of that sphere, and then have those tracks grow through this reprojected 2D, like, 2D planes in time, which makes a 3D object, which then you have all the tracks and you see actually sort of the tracks move, and you gain insight from those groups of cells actually like traveling in 3D and make like this tree sculpture.
Projecting 4D into 3D would be super nice, and that's clearly something you can do in a computer screen. Like 3 to 2, it works, but 4 into 3, like try with the hypercube, have to make it speak to.
It doesn't work. Yeah, I mean, like 4 to 3 is still pretty hard, even though like, it's so complex. But yeah.
We have still one comment from.
I had a quick question. There's one thing that I'm still not able to understand. Even if you do these projections, even if our perception of our data increases by doing these things, we still have to do some statistical validation. Do you imagine there would be new ways to do statistical analysis if our perception has changed with these new Because right now what I see is just in 2D or at the most 3D, but with these techniques you have more dimensions added to your perception. Do you think that statistical methods would still be the same or would they change? And if they are still the same, then why do we need to go to such a complex level to just look at the data in a fancy way?
Yeah, I mean, I don't think like statistics exist sort of like outside the realms of human perceptions in the sense, right? Like it's just like mathematical. You just take your object and you try to like find pattern in that data through uses of math. But what changes to me is more the questions you will ask with stats, right? So like stats is sort of like this older tool set, right? And it has this very, very vast array of questions you can ask with it. But the questions you ask, right, is informed by your knowledge, your prior knowledge, and what your research question is. And you cannot get, maybe you can't get at that research question. Maybe you can't get at that question, right? So it's like happens before the use of stats. That's one. Two, today, you can't get at stats for those things without spending 6 years or maybe making a science, like citizen science game to track cells and motivate people to track cells with like thousands of people to click through things. Like that's the other way, right? Like I'm trying to make this super fast so that, you know, a researcher can do it. But I think, yeah, sure, like making it into a citizen science or Amazon Turk style thing would work as well, right? Like there are other solutions, right? But so
that's like a bird flop game that there's no, you're not, yes, I think that there are interesting scientific questions to ask, but looking how fast people find the right patterns, etc. But it's just going to be really fun. Yeah. Roy had the problem before. Roy?
Yes, sorry, I don't want to keep you for long, but just because the presentation was so engaging, it made me think around a good visualization can help in developing good intuition, but I was thinking about intuition around processes, things like decision-making, things like, and an example which you're talking about, whether it's human oversight, human in the loop, and better understanding of how decisions are getting arrived at, which is not just looking at something at a snapshot, but it's also how the processes evolved and how the group interactions actually happened. And you may be aware of something called swarm AI, which is inspired by exactly flocks of birds and swarm of bees. And what they claim is that they are the best mechanisms to come to an optimal consensual decisions. And the way they do it is by monitoring real-time processes. So I was just thinking, because it's now starting to inform even public policies and things like that. So it's now coming from research stage to in the wild applications. And I was just thinking, because it's making me think around now, when I think of the representation aspect of it, how does it compare with, is that a direction which is interesting to you? Is that something which you have some thoughts around? Like not just looking at a snapshot, means looking at like, the visualization aspect of it, but then also the evolution, real-time evolution of states, both as an observer, as a moderator, as well as a participant.
I mean, the questions the underlying question, right, that motivate this entire thing is the understanding of morphogenesis, which are swarms of cells just, you know, aggregating and disaggregating and forming very complex processes, right? Like this is really the more very fundamental questions that I've drove me to those questions about VR today, right? So like, yeah, I definitely have interest. I don't know that what I do really sort of would be connected to like swarm AI or style things directly. It's sort of connected with like being in a flock of birds and like being the swarm, right? Being the individual in the swarm. And actually what would be even more interesting is if you do this as a multiplayer sort of thing, and then you sort of have like a flux of people and you just put like 200 humans and like you guys have to make a swarm. Right? You have to move together. Not even like a computer anymore. You need to be the swarm. And like you see at the complete breakdown, I think, of the system.
You train them, you invest 1, 2, 5, 10 people in the flock, and then they replace birds, and by the time the game ends, there are no more birds, and there are only people flying together.
Yeah. Yeah. But yeah, thank you.
Thanks, man.
Thank you very much.